Welcome, everyone. I am Sage. Today, we're critically examining Spatial Agent, a paper from Wang, He, Coelho, Bucci, and colleagues at Genentech in Stanford.
Here's our roadmap for today. We'll start by framing the problem in spatial biology, then examine the paper's central claim and how Spatial Agent is architectured. The bulk of our discussion will focus on 4 use cases, with particular attention to the validation approaches. We'll close with critical analysis and open discussion points.
Let's start with why this paper exists. Spatial genomics is a rapidly growing field that generates complex, high-dimensional data, but the computational workflows are highly fragmented. Researchers need to chain together multiple tools, reference databases, and custom code, all requiring deep expertise. The authors argue that LLM-driven autonomous agents can address this by dynamically adapting to new datasets and tasks, unlike rigid automated pipelines. Here is a question for the group. How many of you have experienced these pain points firsthand? Here's the paper's central claim, shown in Figure 1A. Spatial Agent is presented as an LLM-equipped autonomous agent that tackles 4 major tasks in spatial biology: gene panel design for experiments, cell type and tissue niche annotation for optional analysis, finding summarization, and hypothesis generation for cell-cell interactions. The authors emphasize it supports multimodal inputs and cross-species analyses. Note the scope of the claim. This is an ambitious end-to-end system. A good question for the group: is it realistic to expect a single agent architecture to excel across all of these fundamentally different tasks?
Figure 1b shows the 3-module architecture. The memory module maintains both high-level objectives, like what tissue you're studying, and episodic memory of what steps have been completed. The planning module uses chain-of-thought reasoning and can either follow predefined templates or generate plans from scratch. The action module is where the actual computation happens. It selects from 19 specialized tools spanning database queries, data processing, and aggregation. A key architectural decision is the use of predefined templates. The authors acknowledge these templates are critical for stability. Without them, the agent struggles with complex tasks like spatial gene regulatory network inference. This raises the question: how much of Spatial Agent's success comes from the LLM reasoning versus the carefully engineered templates?
Let's examine what's under the hood. Spatial Agent integrates 5 databases, including CZI CellXGene with over 135 million cell profiles, plus PengLaoDB, CellMarker2, and others. On the processing side, it leverages established libraries, ScanPy for preprocessing. A key observation: much of this is wrapping existing, well-validated tools rather than creating new computational methods. The novelty is in the orchestration layer.
Now, let's deep dive into use case 1, gene panel design. The task is to select an optimal set of genes for spatial transcriptomics experiments on the dorsolateral prefrontal cortex. Figure 2a shows the agent's workflow. It retrieves its template, queries multiple reference datasets in parallel, searches PengLaoDB and CellMarker2, assigns importance scores to genes, then aggregates across multiple runs for higher confidence. Notice the template-guided nature. This isn't the agent reasoning from scratch. It's following a structured playbook and dynamically adjusting. They benchmark against 4 computational methods and 10 human experts, which is a commendably thorough validation setup.
The quantitative results are shown here. Spatial Agent consistently outperforms the 4 computational baselines across panel sizes from 50 to 500 genes. The improvements are most pronounced at smaller panel sizes: 6 to 19% better cell type prediction accuracy and up to 47% R-squared gains in spatial coordinate prediction over methods like PERSIST. Notably, the advantage narrows at larger panel sizes, which makes biological sense. With 500 or more genes, there's enough coverage that most methods converge. The authors provide pairwise t-tests in the supplement showing statistical significance at 50 genes. A fair critique: HVG performs nearly on par with Spatial Agent for spatial prediction, with only a 1.4% difference, suggesting simpler methods can be competitive for certain metrics. Perhaps the most impressive validation is the head-to-head comparison with 10 human scientists. Spatial Agent beat 90% of human designs in cell type prediction and 95% in spatial coordinate prediction. But what I find most interesting is the hybrid result. When the agent refines human-designed panels, it improved cell type accuracy in 80% of cases and spatial prediction in 90%. Remarkably, 55% of these hybrid designs outperformed the agent working alone. This suggests genuine complementarity. The humans bring domain insight that the agent lacks, and vice versa. The maximum improvement was a striking 935% in predicting y-coordinates. Here is a question for the group. Does this hybrid result change how you think about deploying such tools, as replacements or as collaborators?
A key strength of this paper is the interpretability analysis in Figure 3. The agent doesn't just select genes, it provides natural language rationales for each selection by integrating multiple knowledge sources. Genes corroborated by multiple databases receive higher confidence scores. The top 3 genes selected, GAD2, SLC17A7, and GRIN2B, are well-established neuronal markers with known roles in the DLPFC. In contrast, SpApros selected genes like PCDH15, ATP1A2, and NKAIN3, which show weaker spatial enrichment. The clustering metrics also favor SpatialAgent across all 4 measures. But here's a critical question. Is the agent genuinely reasoning about biology, or is it pattern matching from its training data on well-studied tissues like the DLPFC?
Use Case 2 tackles cell type and tissue niche annotation on a complex developing human heart dataset. This is a more challenging task than the DLPFC because developing heart tissue contains diverse cell types along continuums rather than discrete categories. The figure shows the workflow and comparison across methods. SpatialAgent integrates multimodal information, Murfish expression data, plus anatomical reference images, which gives it an edge over gene-only approaches like GPT cell type and cell typist. A critical methodological note, the authors use the original paper's author-provided annotations as ground truth, which they acknowledge introduces inherent bias. This is a common problem in the field, but it's worth discussing whether this is a fair benchmark. Looking at the quantitative results, SpatialAgent achieved about 82% accuracy for cell type annotation, closely matching the best human expert. GPT cell type was slightly behind at 81%, but cell typist performed poorly at only 46%, largely because it was designed for scRNA-seq rather than Murfish data. For tissue niche annotation, there were no existing automated baselines, making SpatialAgent arguably the first tool for this task. It matched or outperformed all 7 human annotators while completing the analysis in about 30 minutes versus hours for humans. However, let's note what SpatialAgent got wrong. It failed to identify epicardial cells, a tissue-specific population that some human experts caught. This reveals a key LLM bias, defaulting to common annotations from training data rather than incorporating dataset-specific context.
This is one of the most illuminating examples in the paper. Leiden cluster 18 from the heart Murfish data was annotated as cardiac fibroblasts by GPT cell type, because the top differentially expressed genes included extracellular matrix genes like postin, COL2A1, and COL9A2. A purely gene-centric approach was misled by these markers. SpatialAgent, in contrast, combined transferred cell type composition information, finding a 54% neuronal proportion, with neuronal markers like NRXN1, and correctly identified the cluster as neurons, matching the author's annotation. Crucially, SpatialAgent also flagged the overlap with cardiac genes as a potential caveat. This is exactly the kind of transparent, multi-source reasoning that distinguishes agentic approaches from simple LLM prompting. Here is a question. How often do we see this kind of misannotation in practice with single-modality approaches?
One of the more novel contributions in this paper is the collective intelligence approach for tissue niche annotation. When SpatialAgent annotates individual samples, it makes errors due to limitations in LLM visual reasoning, for example, mislabeling the right atrium as the tricuspid valve. But when it aggregates reasoning across all samples simultaneously, these inconsistencies get resolved through consensus. This is clever engineering, but note the critical caveat. The authors themselves attribute the initial errors to fundamental limitations of visual reasoning in current LLMs. The cross-sample aggregation is a workaround, not a fix. A good discussion question: what happens when the majority of samples have the same error? The consensus approach would amplify rather than correct it.
Use Case 3 is perhaps the most ambitious. Open-ended discovery on a dataset published after GPT-4o's knowledge cutoff, which is an important methodological choice to avoid knowledge contamination. The agent autonomously ran a multi-step analysis pipeline on a DSS-induced mouse colitis dataset, summarizing condition-specific cell compositions, computing cell-cell communication scores using Liana Plus, performing factor analysis, and generating a comprehensive 7,000-word report. The interaction mode shown here allows users to ask follow-up questions and launch new analyses via memory hacking. This is where the agentic paradigm really distinguishes itself from simple prompting. The system maintains context across a complex multi-step workflow. Figure 6b is a fascinating comparison. SpatialAgent independently reproduced key findings from the original study, notably the role of inflammation-associated fibroblasts and significant immune infiltration. But it also had insights that were emphasized in the original paper: enhanced TGF-beta signaling, submucosal remodeling, and fibroblast-parasite interactions suggesting ECM remodeling. The agent proposes that fibroblast polarization, mediated through TGF-beta and IL-11 signaling, is a critical process in tissue regeneration. These are plausible biological hypotheses supported by prior literature on IL-11 and fibrosis. However, a critical point: these novel findings remain computational hypotheses. The paper does not include any wet lab validation of these specific predictions. How should we evaluate the novelty of computationally generated hypotheses that haven't been experimentally tested?
The fourth use case is particularly relevant for practical adoption. SpatialAgent was used in an ongoing wet lab experiment to design a customized gene panel. They tasked it with selecting 100 additional genes to enhance the Actinium 5K pan-tissue panel for prostate cancer mouse models. The selected genes improved the definition of basal epithelial cells, fibroblasts, and prostate microvascular endothelial cells. Crucially, the enhanced panel also revealed laminin-integrin signaling networks that were completely undetected with the standard panel. The authors show that the Actinium-only panel primarily identified fibroblast immune interactions involving JAG1 Notch2 and VCAN TLR2, while the enhanced panel highlighted previously undetected laminin-integrin networks. This is the closest the paper comes to real-world wet lab integration, though the wet lab data collection was still in progress at publication time.
Zooming into the cell-cell interaction analysis, the enhanced panel revealed cell-type-specific communication patterns that the standard panel missed. Fibroblasts engaged in numerous robust interactions across immune subtypes, while basal epithelial cells displayed fewer but functionally important integrin-based signals. The discovery of IGF-1 INSR signaling specifically in Tregs is particularly interesting from a translational perspective. However, the authors note these results are from benchmarking against a reference scRNA-seq dataset, not from the actual Xenium spatial experiment, which was still being collected. This is a significant caveat. The spatial validation that would truly demonstrate the panel's value was incomplete at publication.
Let's step back and evaluate the overall validation strategy. This table summarizes what was validated and what wasn't across each use case. Gene panel design has the strongest validation: 4 computational baselines, 10 human experts, and hybrid evaluation. Cell annotation is also well validated against 2 automated methods and 7 experts, though relying on author annotations as ground truth is a limitation. The CCI mining task is validated only against the original study's findings, with no head-to-head computational baselines. And the Xenium customization has the weakest validation, benchmarked only against reference scRNA-seq data, with wet lab results still pending. Notably, none of the novel biological hypotheses generated by SpatialAgent have been experimentally tested. This is a significant gap for a paper claiming to enable hypothesis generation. Now, let's put on our critical hats. I want to highlight 5 key methodological concerns. First, template dependency. The authors acknowledge that the agent failed on spatial gene regulatory network inference when no template was available. This raises the question of how autonomous the system truly is. Following a template is more akin to executing a protocol than genuine scientific reasoning. Second, the ground truth problem. Author annotations are used as gold standard, but some human experts actually identified structures the authors missed, like the epicardium label. Third, LLM knowledge bias. Both SpatialAgent and GPT cell type failed to identify epicardial cells, even when they were explicitly mentioned in the instructions.
This suggests LLMs default to the most common annotations from training data, which is problematic for rare or tissue-specific cell types. Fourth, reproducibility. LLM outputs are inherently stochastic, yet the paper provides limited characterization of run-to-run variants. Fifth, and most fundamentally, the novelty is in orchestration rather than new methods. Is that enough to claim a paradigm shift? Let me highlight 5 specific things the paper does not address. First, there's no uncertainty quantification. The authors themselves flag this in the discussion as crucial for trust, but it's absent from the current system. Second, there's no ablation study. We don't know whether the performance comes primarily from the templates, the tool selection, the multi-source aggregation, or the LLM reasoning itself. Third, only 3 tissues and 2 species were tested. For a system claiming general-purpose spatial biology capability, this is narrow. Fourth, the hallucination risk is acknowledged in the discussion, but never quantified. How often does SpatialAgent make biological claims that are simply wrong? Fifth, none of the novel hypotheses have been experimentally validated. These are limitations, not fatal flaws, but they constrain how far we should take the paper's claims. A good discussion point. Which of these would you most want to see addressed in a follow-up study?
Let's wrap up with broader implications. The democratization potential is real. SpatialAgent could empower labs that lack computational specialists to perform state-of-the-art spatial analysis. The cost savings are dramatic. But there's a fundamental tension between autonomy and oversight. Who validates the AI-generated hypotheses before committing wet lab resources? The authors envision a future of closed-loop experimental design, where the agent proposes experiments, the lab tests them, and the agent refines. That's exciting, but requires a level of trust that the current validation doesn't fully support. I'd also flag a practical concern. The system depends on GPT-4o's API, which could change, be deprecated, or have its knowledge cutoff shift. How do you version control science that depends on a commercial API? Let me pose 5 discussion questions to the group.
So, what's our verdict? I'd say the gene panel design and human-agent collaboration claims are well supported. The validation is rigorous and the results are convincing. The cell avitation and CCI mining claims are moderately supported. The results are promising but limited by ground truth bias and lack of experimental validation, respectively. The overarching claim of a paradigm shift is premature. SpatialAgent is clearly a useful tool that can accelerate spatial biology workflows, and the hybrid collaboration results are genuinely exciting. But a paradigm shift requires broader validation across more tissues, species, and experimental settings, plus experimental confirmation of the novel hypotheses. I'd encourage the authors to pursue the wet lab validation of their colitis findings and expand to less well-studied biological systems where LLM knowledge is weaker. What does the group think? Are you convinced this is more than a well-engineered wrapper? Here are all the references cited throughout this presentation. The primary paper is the Wang et al. BioRxiv preprint from April 2025. The supporting references span the spatial biology datasets used for benchmarking, the cell-cell interaction literature that contextualizes the colitis findings, and the broader AI agents field.
Thank you for your attention. To summarize, SpatialAgent demonstrates that LLM-based autonomous agents can genuinely accelerate spatial biology workflows and collaborate productively with human experts. The hybrid collaboration results are the most compelling finding. However, the paradigm shift claim needs stronger support through broader validation, uncertainty quantification, and experimental testing of the novel hypotheses. I'd love to hear your thoughts. Who wants to start the discussion?